Our first speaker is Luca Pandolfini, who studies environmental sex determination in turtles. He does that at the Italian Institute of Technology. Luca, take the stage, please.
So, hello everyone, and I want to thank the ONT team for giving me the chance to present my data. So today we are going to talk about this little fella, the tiny tortoise Pyxis arachnoides, which is one of the smallest tortoises in the world, which is unfortunately critically endangered by both his low reproductive rate and by increased threat to its natural habitat. This tortoise comes from southwestern coastal regions of Madagascar, and from a very important biodiversity hotspot. This is the so-called spiny forest, xerophytic environment, which is threatened by a lot of man-related activities, and therefore its population has been depleted starting from the 90s to near to extinction rate. Unfortunately, though, we need to take actions in order to preserve this organism. In order to take action with conservation strategies, the determination of sex and parental lineages of a species are of paramount importance. And nanopore devices, being portable, are really a good asset to these kind of conservation campaigns. Tortoises, as most reptiles, are characterized by environmental sex determination. Therefore, is a non-chromosomal sex determination mechanism, following the rule of hot girls and cool guys, in the sense that at warmer temperatures you have a higher chance of getting female, and at lower temperature of getting males. But unfortunately, especially in very young individuals, the confirmation of their sex is very traumatic and invasive. On the other hand, reptiles have nucleated blood cells, so you have easy access to the genetic material. So by a combination of 110 and 114 reads, and also by POR-C, using the latest KIT14 sequencing kits, we were able to collect from 50 to 100 hex coverage of the genomes of this organism. And we put in place a pipeline of assembly by using NeXT-DeNovo, NeXT-Polish, and 3D DNA canonical assembly that allowed us to get chromosome-level reference genomes in less than 4 months. It's really amazing. And by all means, all qualitative parameters of the genome we obtained was in line and comparable with what you can obtain by standard Illumina or PacBio-based reference genomes for other turtle species. And also the synteny was very well in line with already available turtles genomes. And finally, we were able to identify a few hundred of high-confidence promoter-associated differentially methylated regions that constitute a footprint of what distinguishes from an epigenetic point of view a male from a female tortoise. So next step is to adapt this strategy and scale it down to very shallow sequencing devices in order to be portable in the field. I want to thank you, all the collaborators from Aquario, University of Zurich, and Org-One for giving me the opportunity to carry on this project, and also you for your attention. Thank you.
Yeah. Thank you, Luca. Our next panelist is Matt Miller from Reneko International Wildlife Consultants. Matt's going to tell you about his work, some of the world's largest conservation projects.
Yeah, hi. Thank you very much for inviting me here to speak. I'm really excited to be here. I work for Reneko Wildlife, International Wildlife Consultants, and we run the largest vertebrate captive breeding program in the world out of UAE. We breed various birds that are part of the traditional Arabian falconry. And so you had birds like the Hubara bustard, which in the 1950s through 1980s suffered a very severe decline and was at risk of going extinct. And through the leadership of the Abu Dhabi government, they've invested quite strongly in a captive breeding program, which has resulted in a lot of basic science, including genomics work. And so I'm going to talk a little bit about how our genomics work has informed that stuff and where we're moving forward with it. So what we recognized is that for us, in order for us to manage birds like the Hubara in their native range, we had to understand their population structure. We had to understand something about their demography. We had to understand how inbred they were, which meant that we needed to do genomics, which meant we needed a reference genome. And typically, reference genomes are these very difficult affairs. Increasingly, over the last few months, it's becoming more and more easy. But in fact, we're thinking that we can do this even easier and faster at scale. So I want to point out that every genome that you've ever looked at is a model. And just like a map, no genome is perfect, right? And so I showed here a map from the 1500s of the New World, and you can see just how wrong it was. But this map was good enough to allow navigators to cross oceans and to successfully explore the New World region. And in the same way, these genomes that are good enough we can use to understand genomics, especially from a biodiversity standpoint, because what we're interested in is large contigs of ungapped sequence so that we can do various demographic and inbreeding analyses. So here's an example of 4 genomes that we've worked on recently, and what I want you to see in there is we have very high BUSCO scores, and we have extremely long ungapped contig N50s, okay? And you'll see there's 2 birds there and 2 fish there, and that's really important, the fish, because this is how we're pivoting. So in fact, you'll see that we had about 30x coverage, and these are very good genomes, and they're not at chromosome. We could make them to chromosome. You can always improve your genome, but these are very good, and we were able to generate these genomes from sequencing to draft genome in a week or less with a team of 2 or 3 people. And these cost, in terms of consumables, at least 1/3 of what a traditional platinum level genome would cost. So this allows us to do it at scale. And in fact, the thing that I want to point out here is, and this is where that circle chart is there, our genomes, although not technically classified as chromosome, have longer fragments. If you take a fragment of 30 million bases ungapped as our N50, only 6% of bird chromosome level genomes achieve that metric. And so ours are able to do that in a very cheap way. This scales, and in fact, what we're trying to do is to sequence all of the native fish genomes from the Red Sea. And the only way we can do that at scale and quickly over the next 3 years is by taking this fast approach. So if you have any more questions, see me about how to do this. I'd be very happy to talk about how you guys can also do genomes good enough for conservation.
Thank you, well done.
Thank you, Matt. Our third panelist is Phoebe Aldag, who works for Basecamp Research. Occasionally, science and the world of adventure overlap, and I'll leave Phoebe to tell you about that.
Hi there. So I'm maybe going to start with something that this audience doesn't need hammered home to them, which is the importance of proteins. So I think without the discovery of Taq polymerase, we'd all still be sitting adding new polymerase into our PCR machines at every cycle. And so that same transformation that thermostability provided for the molecular biology fields, new proteins are offering to medicines or therapeutics, which are the same thing, or materials or food industries. And so Basecamp Research is trying to work on this. It's a really exciting field, but it's a bit of a wild west where these proteins are coming from. And there's 3 major problems with the protein sourcing that's happening right now. One is that they're not coming from a representative sample of the genomes out there. So there's an estimated 1 trillion species on this planet Earth. And in NCBI, 2/3 of all of the sequencing reads come from 12 species. And I bet you can guess some of the heavy hitters there. So number 2, and maybe most importantly, they're not meaningfully linked back to the biodiversity sites from which these proteins have come. And third, they're dropped into sort of categorical lists and some of the really important relationships are lost. And so what Basecamp Research is doing differently is building a global supply chain from all biomes, and really importantly, making a link between the biodiversity guardians and the biodiversity hotspots and the biotechnology industry. So we're guided by the Nagoya Protocol. Any commercial success that we find, we share back in the form of royalties and in the form of data sharing and in the form of capacity building. And so finally, I think biologists intuitively understand the importance of networks, from food webs to gene networks. And so we're really excited that we're organizing all of our biological data in a graph database and applying novel graph algorithms to really understand and drive insights around function that otherwise we might miss. So this global supply chain is something we're really excited about, and we have a field team, which I'm leading, which is comprised of Antarctic ice divers and experts in off-grid molecular biology, sort of the adventure that Ian was alluding to. So I think we're really nailing it on the anything, anywhere, but what we're excited about moving forward is changing this focus to anyone. So instead of our field team helicoptering around the world, we're building up partner labs in country with the people who are actually there guarding this biodiversity, to have them join us on the exploration and hopefully find novel proteins, but also develop more of an understanding of the biodiversity that they are taking care of. And so we're really excited about how far we've come thus far. These slides were submitted in April, so they're out of date already. But as of April, we had 68 expeditions across 19 countries that have covered over 40% of the world's biomes. And this has led to our graph database having over 2 billion relationships, and in terms that maybe make more sense, that's 4 times the diversity of NCBI and 3 times the size of UniProt. And thanks to the long reads of Nanopore, our assemblies are 5 times longer than those on Magnify. So we're really excited about this. This might have come across a bit like an ad for Basecamp, and I hope instead it can be an ad for nature, and the concept that so many global problems are out there that nature, 4 billion years of evolution, have already solved. And so hopefully we can work together to find the answers. Thank you.
Thank you, Phoebe. Love to get some questions from the floor for the panelists. While you're thinking of questions, and I'll ask the first question of the panel. So I think all 3 of your projects and the work that you do are completely, they're radically different from each other, and each in its own right, I think, unique. And I wanted to ask you about that, the novelty of the work that you do, and it's paradigm-changing work that you're doing, and how much of a motivation that is to you in your daily work, how important that is to you in the projects that you do. Does anybody want to take that one, Phoebe?
Yeah, I would love to, thank you. I think there's kind of 2 elements to this. One is that bioprospecting, looking for solutions in nature, has been happening forever. So many of our antibiotics already come from soil microbes. So in some way, we're really doing the same thing people have done before, but we're trying to change the narrative by making access and benefit sharing central to it. And so, yeah, I think we're sort of building upon what's already been there, and that's really important to not reinvent the wheel, but improve upon what's out there.
Yeah, great, thanks. Either of you like to take that question also?
Well, so from our perspective, we are always interested in looking for, again, ways that our genomic data can actually influence decisions. And our pivot to the Red Sea, which has been quite recent, is motivated, obviously, my personal interests are to discover biodiversity. And I love to characterize biodiversity. I have a long history of being a field scientist, and I'm enraptured by biodiversity. At the same point in time, we all know that this is challenging. How do we fund this? And so much of the motivation in the Red Sea Fish Program is actually motivated by looking for aquaculture solutions in a sea that is both very hot and very saline. And so how can we get fish that can survive, thrive in that, and provide a protein source for humans in a growing population in the Middle East? And so we think using native species, right? And so this is the sort of win-win approach that is guiding our work in terms of advancing biodiversity discovery, but also finding a motivation that is admired in human progress and development.
Yeah, great, thanks. And Luca?
So in what was more remarkable to me, and I think has the highest impact in conservation biology by using this technology is that virtually breaks the divide between different fields that have to come together in order to carry on a conservation effort. In fact, in the past you had a big sequencing effort in order to characterize the genome, then another divide was between the field work and the lab work. You usually collected samples and then you went back and have a look at what you got, and then you have an answer, but maybe it's too late because you are already out of the field. You virtually can do everything all together, or at least much easier straight in the field with this technology, and also the time required from going from I don't know anything about this species and I know even the epigenome of this species is really remarkable. And also in the time frame that takes to go from one point to the other is months, not years as in the past. So.
Yeah, great. Thanks, Luca. Does anybody have a question for the panel? Any of our panelists?
Hi, I have a question for Matthew. So you're sequencing birds and fish, which are the most lucky samples we can ever sequence in Nanopore. Can you comment on it? Can you tell us what are your tips and tricks?
I suffer from the same issues that you do. We have blocky DNA. And with our wonderful team here from the UAE, Divin is our technical guru, we've been trying a few things. Unfortunately, I can't tell you we've had a ton of success. I can tell you a few things. Bird DNA is blocky in a different way than marine fish. It just behaves differently. They're both blocky, but they're not the same. You know, bird dies quickly and you can't recover the flow cell. Marine fish also dies quickly, maybe even more quickly, but with a flow cell wash, you get the flow cell back. So it's very different. What we've been trying with birds and what I'm excited by is heat shock and increased fragmentation. The heat shock was Divin's idea to sort of knock off, you know, some structuring elements that might work. We seem to have some evidence that that's going to work, but we're hoping to partner more closely with Nanopore on this issue. On the other hand, we're finding that we don't need huge coverage. For bird genome assemblies, because they're not so repetitive, if we have 20x coverage, we can assemble a genome. So quite candidly, with almost every organ, yeah, I know, it's crazy, but this is this new chemistry, right? I mean, really, it makes a big difference. And we've also been improving our pipelines, and I'd be happy to talk to you about what we're doing on the bioinformatic end to wrestle as much data out of it. But in general, we actually think with a MinION flow cell, we can do most bony fish because their genomes are less than a gigabase. And for most birds with one Promethean flow cell, and you'll note that Oxford Nanopore just lowered the price of the Promethean flow cells. So that makes getting your favorite bird's genome even more accessible to your team.
Thank you.
Thanks. Any more questions from the floor? Okay, I have another question for the panel. So you've all talked about sequencing DNA. You're 4 times the size of NCBI reference genomes, but you're looking at methylome. And what do you think about applying Nanopore sequencing beyond genomes and beyond reference genomes? New types of data and how that will impact conservation efforts. Who would like to take? You want to try and look at?
So, yeah, for us, the main struggle was to find the right tools, bioinformatics tools. So I would say that the 4-5 months that it took from beginning to the end, half of that time was to find the right tools that really were achieving for us the best QCs in terms of genome contiguity, mesoassembly was reduced to the minimum. So in our hands, we were quite perhaps lucky to find the right condition to get a lot of data. And despite being reptile samples, they were not that hard to sequence, even with quite high N50s with canonical ligation-based sequencing. But on the other hand, it took weeks and weeks of different tweaking with the tools to find the right pipeline to achieve comparable, if not better, quality to compared to other reference genomes.
Can I take a stab at this? So I'm going to share with you guys what I think is most exciting. So we do these reference genomes. And if you've done a reference genome and you're interested in population genomics, you're probably still using Illumina for your resequencing project. So at the end of the day, you need population data. And right now, seems like the best way forward is Illumina. But that involves all sorts of problems when you're working around the world, when you don't have access to an Illumina sequencing center. Sending your sample, permits, it's also this sort of, we've heard several times today, this sort of helicopter approach to doing research that's sort of colonial and not what any of us want to do. It doesn't really empower local labs. Doesn't work. So what I'm really excited about, and since we first started talking with Nanopore back at the PAG 30 conference in January, is how we can do low-pass imputation to remove Illumina from our data set completely. And I was really excited by Dan's talk about this topic because it's something that we in our lab have been trying to solve sort of on our own. With how do you do variant calling? How do you deal with it? The version 14 chemistry gets us to the point where it's reasonable and feasible. And if you just run your math, again, I'm going to use a bird example, I can probably do a 1x coverage genome for a bird for about $33 in consumable costs. Now that's pretty good, and that beats the pants off of anything I can do on Illumina. I don't have to send it to a center, and I don't have to wait weeks for my data. I can actually genotype in the field. And for endangered species, this is the holy grail. And so this is what I'm super excited about in this space.
Beats the pants off anything I can do with Illumina. That's my favorite sentence of the day. Phoebe, just a few seconds.
Yeah, I have a slightly
beyond genomes.
different take to Matthew. That's amazing. I'm also really excited about the other modalities that Nanopore is developing and offering. So the end goal for Basecamp is to find proteins that we can license out to the food industry or the medicine industry or whoever. And for them, it's really impactful if we're able to give more information about the protein. So being able to infer things like the expressibility, maybe if there's cofactors, what pH range or temperature gradients these things work at is really, really helpful. And so to have transcriptomic data really helps us to infer more about the protein function. And my dream is direct protein sequencing that could also read out PTMs, because that will just help us so much in understanding why won't this express when I throw it into a totally unrelated host.
Okay, great. Thank you so much. Thank you very much.